Platypus Technologies has developed a unique method for performing cell migration assays without the use of transwell membrane inserts. This unique assay uses a 96-well plate populated with Oris cell seeding stoppers. The tips of these stoppers create exclusion zones in the well, and cells will adhere around the tips. When the stopper is removed from the well, the cells can then migrate into the center of the well. Migration can be detected via microscopic analysis, digital imaging, or with a fluorescent plate reader. To begin the assay, if you will be quantitating the cell migration using a fluorescence plate reader, snap on the detection mask to the bottom of the plate prior to seeding your cells. This detection mask allows only the fluorescence from the cells that have migrated into the center of the well to be detected. The mask is not required if the results will be viewed microscopically. The Oris Cell Migration Assay has been designed to work with adherent cell lines, such as HT1080 fibrosarcomas, 3T3 Swiss albino fibroblasts, and HCEC epithelials. Allow cells to reach 80% confluency, harvest, and then aliquot 100 microliters of cells into each well. The optimal number of cells will be dependent upon your specific cell line. Lightly tap the plate on your work surface to evenly distribute the well contents. At this point, cells can be labeled with a fluorescent reagent or express fluorescence via the green fluorescent protein. Incubate the cells overnight to allow for adherence to the well surface. Once the cells have adhered, use the Oris stopper tool to remove the cell seeding stoppers, thus creating the detection zone in the center of the well. It is important to lift the tool vertically and not to use the stopper tool as a lever to pry the stoppers from the well. For reference purposes, leave several stoppers in place in order to establish time equals 0 for the migration process. Incubate the cells to allow migration to occur. Due to the flexibility of the Oris assay design, migration can be detected via microscopic analysis, digital imaging, or with a fluorescent plate reader. Here you see an image of pre-migration, time equals 0, where the exclusion zone created by the stopper is visible. In this image, you can now see the cell migration that has occurred in the detection zone. In addition, both kinetic and endpoint cell migration analyses can be performed. For kinetic assays, multiple readings can be taken over various time points during the migration process. For endpoint assays, simply read results at the end of the incubation period. A variety of stains and labels can be used to detect cell migration in real time using the Oris assay design. The Oris Cell Migration Assay is an ideal alternative to wound healing and scratch assays. Unlike transwell membrane inserts, the Oris assay design allows researchers to visualize cell movement in real time while offering the ability to quantitate results.